Welcome once again at Medical Sciences by Naphtali Muhumuza. And in this session, we are going to discuss on the anatomy and physiology of the duodenum. Duodenum is the first part of the small intestine, whereby we know that the small intestine is divided into three. That is the C-shaped duodenum. We have the jejunum and the ileum. And we know that duodenum being the first part is what we want to start with. Then in the next video, we shall talk about the ileum. And this duodenum is supplied by accessory organs of the digestive system, and that is the pancreas and the liver. So we are going to see the interplay of the liver and the duodenum and the pancreas playing a big role. So let us get started. So we are saying that this duodenum is C-shaped, it is C-shaped, and apart from being C-shaped, it is the peritoneal, it is the retroperitoneal, meaning it is found, retro means outside, it is found outside the peritoneum, and it is C-shaped, and we have said it is the first part of the small intestines. And this duodenum, we have said it has accessory organs, that is the pancreas and the liver. All of them meet at the duodenum. And the junction where they meet, we call it ampulla of Vater. The junction where the common bile duct meets together with the ducts of the pancreas, we call it ampulla of Vater. Ampulla of Vater, or you can call it hepatopancreatal ampulla. That is where they meet. So that whenever the secretions from the gallbladder and the liver come in, they meet with the secretions of the pancreas at the ampulla of Vater, what we call hepatopancreatal ampulla. And this duodenum, we see it is the one that is connecting the jejunum here together with the pylorus part of the stomach, whereby what enables the food chain to enter into the duodenum, they pass via the pyloric sphincter. It is the one that enables food to enter. And this pancreas, which is an accessory organ, it is divided into three, whereby it has the head, the body, and the tail. Then we have seen also the liver, which this is the falciform ligament, and this liver is divided into the left lobe and the right lobe, together with the bile duct, the gallbladder. And this gallbladder empties into the bile duct via the cystic duct and also brings into the ampulla of Vater. So if there is a problem with the liver, a problem with the pancreas, you can also see a problem with the duodenum. And this duodenum lining, the mucosal lining of the duodenum, the epithelia, we saw it was simple columnar, and this simple columnar, it has different cells, whereby the first cell, it has the CCK cells, it has the S cells, it has the K cells, and apart from these cells, enteroendocrine cells, it also has Brunner's gland, and in its brush borders, in the brush borders of the duodenum, we saw also production of enterokinase enzyme. Enterokinase enzyme, which we shall see that this enterokinase is the one that activates trypsinogen to trypsin. It activates trypsinogen to trypsin. That is the work of enterokinase. Then the Brunner's gland, for them, they produce bicarbonate ions. Remember, the food which is upcoming is acidic, so it is neutralized by the bicarbonate ions produced by the Brunner's gland. So these ones make the environment become alkaline, creates the alkaline medium. That is the bicarbonates produced in the mucus from the Brunner's gland. Because the Brunner's gland is producing the mucus, and this mucus contains bicarbonate ions that neutralizes the acidic pH and provide alkaline linkage. Then we see also these CCK cells producing cholecystokinin, prothymine hormone, CCK hormone, and we shall see S cells producing secretin hormone, and these K cells, for them, they produce gastrointestinal peptide hormone, whereby these, the one from S cells and K cells, these ones inhibit, they act by inhibiting gastrin hormone production, they inhibit production of gastrin hormone, whereas CCK, it does two roles, whereby it goes and stands on the gallbladder and causes secretion of bile. So we see it stimulates secretion of bile containing bile salts. Then it also goes to the pancreas and stimulates the cells of the pancreas known as acina cells. Because the pancreas, the histology of the pancreas, apart from divided into three regions, that's the head, the body, and the tail, it contains two types of cells. And these two types of cells produced by the pancreas, one of them is called acina cells. And these acina cells make up 99% of the cells of the pancreas. Then the 1% is by the islets of Langerhans. Islets of Langerhans, whereby these islets of Langerhans, they contribute 1% of the cells of the pancreas. So we are saying that cholecystokinin hormone, apart from triggering the gallbladder to release bile containing bile salts, it also goes to the acina cells of the pancreas and stimulates the acina cells to produce pancreatic juice. And we shall see composition of the pancreatic juice. So what it triggers is that when acidic food chain reaches on the CCK cells, it stimulates them to produce cholecystokinin hormone, which triggers production of the bile from the gallbladder and contains bile salts, the sodium taurocholate. And then we see also production of the pancreatic juice from the acina cells. These acina cells, they are exocrine. Whereas islets, they are endocrine cells. That's why we say the pancreas plays both exocrine and endocrine role. Whereby this pancreatic juice, after being stimulated by cholecystokinin hormone, it is produced majorly in two ducts. The major one is pancreatic duct, which opens at the ampulla of Vater, and another one is accessory duct. Or you can call it duct of Santorini. So these are the two major ducts where pancreatic juice passes to be emptied within the duodenum. And we see that these ducts, they are regulated, their secretions are regulated by the sphincter known as sphincter of Oddi. Sphincter of Oddi, I'm not meaning. So sphincter of Oddi, it is the one that regulates the secretion of pancreatic juice via these ducts. We call it the sphincter of Oddi. So that is the function of CCK, production of bile. And what is the function of bile? This bile is produced because it contains bile salts, and these salts causes emulsification of fats. That whenever the food contains fats, these fats are very big that they cannot be digested by the pancreatic enzyme. That is pancreatic lipase. So what the bile salts do, they come and solubilize, whereby they come and create powers into this big fatty globule and break it into small droplets known as micelles. And these micelles, when they are created, they increase the surface area for action of pancreatic lipase and other pancreatic enzymes. So this is the role of the bile salts, emulsifying or solubilizing big fatty globule into smaller droplets, micelles, to create surface area for absorption. Then we have seen the function of this, we have seen the function of this, we have seen the function of bicarbonate creating alkaline pH. Then the next thing we have to talk about is the composition of pancreatic juice. What do we find in the pancreatic juice? And this pancreatic juice, which is produced, contains 99.5% is water, then 0.5% is solutes. And these solutes within the pancreatic juice, we find majorly electrolytes and enzymes. Electrolytes and enzymes, whereby electrolytes, in here they are the sodium, we find the electrolytes being there. We have electrolytes, they are different electrolytes. Then we find their enzymes. And allow me to split these enzymes into two. These enzymes, let me bring them here. These enzymes in the pancreatic juice, we have enzymes, we have the proteases, those which work on proteins, and we have those ones which work on lipids, major. And sometimes we have also the ones which work on nucleic acids. So the enzymes which work on proteins, one of them, they are produced in inactive form. We have trypsinogen, we have chymotrypsin, chymotrypsinogen, we have elastase. Actually, it is produced in inactive form as proelastase. So these are the major ones working on the proteins within the pancreas. And these ones are produced in inactive form. And what happens is what activates trypsinogen is enterokinase. So the enterokinase produced by the brush borders of the duodenum comes and activates trypsinogen. So trypsinogen is activated by enterokinase enzyme produced by the brush borders, and is activated trypsin. And after being activated, it says, since they have helped me, I also have to help others, as God says also. So that whenever you are helped, always also make sure to help others. For example, when you are helped with this video, please help others by sharing it, so that many should come to know about it. So this trypsin, which is activated by enterokinase, is the one that comes and activates the rest. So we see chymotrypsinogen being activated by trypsin and the proelastase being activated by trypsin to elastase. That is how they are activated. And after being activated, we know trypsin acts on the peptides, whereby small peptides which were created by pepsin are further broken down by the trypsin to form tripeptides and dipeptides. Then we see the chymotrypsin also working on the carboxyl ends of the amino acids, breaking the peptide bonds at the carboxyl ends. Then the proelastase also works on the aromatic amino acids, breaking them into dipeptides and tripeptides. That is their role. Then the lipids, the ones that work on lipids, we find enzymes like pancreatic lipase. We find there enzyme like cholesterol esterase, And these ones, for them, they are produced when they are active. They are not zymogens. And we also have phospholipase. Phospholipase A2. This one lipase works on the triglycerides and he hydrolyzes them to monoacyl glycerol plus 2 free fatty acids. Then we see cholesterol converting cholesterol ester. It converts cholesterol esters to free cholesterol. This is the hydrolysis reaction, free cholesterol. Then we see phospholipase working on phospholipids. Because the food we eat contains triglyceride, contains cholesterol esters, contains phospholipids. And we see pancreas, the duodenum being the major site of digestion of lipids. So these ones are working on phospholipids. And they break them down to lysophospholipid. So that is then the nucleic acids, we see enzymes like the nucleases, which works on the nucleic acid. Then the ones which work on the carbohydrates, we should not forget pancreatic amylase. There is also pancreatic amylase, which breaks down the remaining starch. You know, there is salivary amylase, which works on the starch in the mouth. And we saw its digestion ending in the stomach. Then here there is also another pancreatic amylase that breaks the alpha-1 glycosidic bond holding the starch. And also it works on this starch, whereby if I have starch, is broken down to maltose and dextrins and alpha dextrins. That is the action of pancreatic, and all these reactions are hydrolysis reactions. So these are the components we can find in the pancreatic juice. Water being the major component, then 0.5 solutes, whereby we have electrolytes, we have waste products of metabolism, we have enzymes which we have seen. So this is basically what we can talk about in the duodenum. Lastly, we mentioned here that this pancreas, it has two types of cells, the acina, which is the major, and the islets of Langerhans. And these islets of Langerhans, for them, they do great work under endocrinology, whereby these islets of Langerhans, they have three types of cells, whereby they have the alpha cells, they have the beta cells, and they have the delta. They have the delta cells. And these are playing endocrinology role, whereby the alpha cells produce glucagon hormone. These ones, they produce them directly in blood. That's why we are calling them endocrine. Beta cells produce insulin hormone, and the delta cells produce somatostatin. And the somatostatin also plays a big role in digestion, whereby it inhibits production of gastrin hormone. So it also inhibits, and at the long run, it inhibits production of gastric juice. Because without gastrin hormone, no gastric juice. Then we see the insulin doing the role of making the cells to become permeable to glucose. It's the one that enables glucose to enter into cells and tissues. Triggered by hyperglycemia. After eating a carbohydrate meal, glucose goes high, triggers production of insulin. And this insulin is the one that enables glucose to enter into cells. So it acts by lowering blood glucose levels. Then glucagon is antagonistic with insulin, for it acts by increasing blood sugar levels. Increases sugar levels. How? By activating gluconeogenesis, glycogenolysis, and lipolysis. And for it increases sugar levels. So this is the physiology we can talk about in the duodenum. Hope you guys, you have understood something. You have enjoyed the video. Please, if you haven't subscribed, always remember to subscribe. Share to your friends, and your comments are always highly appreciated. Thank you so much.